So, good afternoon, everyone, and welcome to another EV Translational Series talk. I'm Helena Branco, PhD student at University of Porto, Portugal, and together with my colleague Alex Mourinho, a postdoctoral researcher at University of São Paulo, Brazil, I'll be moderating this talk. Today, it is my pleasure to introduce our speaker, Dr. Hector Peinado, Head of the Microenvironment and Metastasis Lab at the Spanish National Cancer Research Center. Dr. Peinado's research focuses on the dynamic communication between tumor cells and their surrounding microenvironment, particularly in the context of metastatic progression. After completing his PhD in Madrid at the Alberto Sol's Institute for Biomedical Research, where he specialized in epithelial to mesenchymal transition, he conducted postdoctoral research at the Weill Cornell Medical College. Since returning to Spain, Dr. Peinado and his team have been leading innovative research aiming to understand the role of tumor-secreted extracellular vesicles in communication with the tumor microenvironment during metastasis and on the potential use of EVs in liquid biopsies. Today, he will present on the influence of tumor-derived EVs in pre-metastatic niche formation, offering an overview of how secreted EVs contribute to pre-metastatic niche formation. Before we start this session, I request the audience to put their questions in the chat box or to unmute themselves and ask the questions at the end of the talk, where we will have a proper Q&A. Now I welcome Dr. Hector Peinado to start this presentation. You may start sharing your screen, and we are looking forward to hear you.
Perfect. Thank you so much. First of all, let me put this at the beginning. So first of all, I would like to thank you guys for the invitation because it's always good to share, the ideas of my laboratory. As Elena mentioned, I started my lab here in Spain in 2015. So we are almost 10 years apart since I started, and I developed these ideas in Spain, and I am actually moving my laboratory to another institution next year. But I will still continue working in the role of the extracellular vesicles in the premetastatic niche formation. So today, let me see. Today, I divided this in five blocks. One is give you an overview of the metastatic evolution, the formation of the premetastatic niche, whether or how to use extracellular vesicles to study premetastatic niche formation. And if you allow me, I put several, let's say, academic approaches in that part, because I think there's many concepts that I would like you to understand on how to approach a project from the practical point of view to study premetastatic niche formation, whether is your expertise in melanoma as me or any other tumor type. So I try to put this more like let's say a specific ideas working in the laboratory and how you can approach it in the laboratory from the practical point of view. Later, in the last part, I put some ideas about the current project we have in the laboratory about trying to understand the EV heterogeneity, and I am going to focus in one molecule that we are looking specifically, which are micropeptides or microproteins. And at the end, I would like to share some ideas about how to use extracellular vesicles for you to be inspired to develop novel therapies. So this is the overview of the talk. And I'm going to start with the metastatic process. As you probably know, you are aware that the metastasis is just a consequence of the tumor evolution. The tumor cell needs to learn how to extravasate in the primary tumor. They need to learn how to survive in circulation. After that, they need to know how to survive to the extravasation and whether you can survive in distal organs. So you should consider this as an evolutionary process. Probably Joan Massaguet, he's inspired us about trying to understand how a tumor cell is able to be not just released to the bloodstream, but actually to home in distal metastatic organs. And around this, my expertise is what in the concept that we know is the premetastatic niche, which are all these changes that are happening before metastasis. So today I'm going to focus my idea on the concept of the premetastatic niche, the mechanistic works that we've been doing and how you can approach this in your laboratory. But before that, let me share also that, we know this complexity, but one thing we were missing actually is whether any cell type within this complex microenvironment is secreting extracellular vesicles. So I think it's important to put in the context of the complexity of the project that you guys are doing, that we are not just formed by cells, but actually extracellular vesicles can play an active role in whether this interaction is happening in the primary tumor, in circulation, or in distal organs. My lab is analyzing the idea or the crosstalk between the tumor and the tumor cells, the tumor microenvironment, sorry. And we believe one of the main, crosstalk that happening is at the primary tumor. I am not working actively in primary tumor, but we are starting to analyze what is the complex interaction between the tumor and the tumor microenvironment in primary tumor. It could be tumor and what is cancer-associated fibroblasts, bone marrow-derived cells, because we know that a big percentage, over 50% of tumors, they carry, tumor cells within a tumor are going to be tumor by 50%, and the other part of the tumor is going to be stroma cell. So we know that the complexity of the primary tumor is not just formed by tumor cell, but we really need to understand the complexity of the microenvironment. Besides these interactions, we learned from our study with David and other laboratories in the world that extracellular vesicles can be shared out in the bloodstream, and they can reach distal organs, in this case, what we call the premetastatic organs. These are organs where the metastasis is going to happen, but at this time, our ideas, when we started this project, we didn't know much about the secretion of the extracellular vesicles at the premetastatic niche and what was the role. So this is how it all started. I am talking about 2005, because David, he published this paper in 2005 where they found that the tumor is secreting what is soluble factors, and these soluble factors were enhancing the formation of what they call the premetastatic niche. In this sense, the premetastatic niche was conceived as a new soil for metastasis. As you know, not all the organs are susceptible to metastasize, and not all the cells will survive in all the organs. So the concept of the premetastatic niche is that this secreted factor by the primary tumor can change those organs, and those organs are different in composition, not only in protein, but also in cell composition, that generate this new microenvironment where the tumor cells can now home and survive much better. This is a question of probability, whether you have an organ that is not preconditioned by any secretive factor. Of course, tumor cells would be able to metastasize in those organs. However, if you are able to form this premetastatic niche, the tumor cells will be able to metastasize much faster and easier. So David started this, and at that time, they saw many other proteins involved in the premetastatic niche formation. They were mostly tumor secretive factors, soluble factors such as SDF, TNF-alpha, TGF-beta. They were all shared out from the primary tumor. They were all somehow preconditioned distal organs, and the main consequences were this: one is a promotion of vascular leakiness. They promote extracellular matrix changes, such as the deposition of fibronectin, tenascin, periostin. There is a lot of extracellular matrix remodeling, and there is inflammation also. So there is a concept that the premetastatic niche is a pro-inflammatory microenvironment, because all those cells that are coming earlier during the premetastatic niche formation are normally bone marrow-derived cells. David saw that specifically bone marrow progenitor cell VEGFR1 positives were one of the first cells getting at the premetastatic niches before any tumor cell getting there. So if you study this, or you have knowledge about the premetastatic niche, you are able to identify those organs of mice which already have a primary tumor but no metastasis, because they already are different. You see several differences among them. So when I started in David's lab, I went there as a postdoc. We didn't know anything about extracellular vesicles, and the idea was just to look specifically how extracellular vesicles can be influencing the premetastatic niche formation. And this is how it all started at David's lab and our research. I joined David's around 2008, and I stayed there until 2014. So during this period, we defined several project. Well, we did several projects that we defined several mechanisms involved at the premetastatic niche formation. Today, I'm going to tell you an overview of what we've been studying, but I don't want to bore you with published data. I rather, as I told you, focus in more practical approaches for you. Maybe later in the discussion point, tell me your view on how to approach these ideas. So as you know, extracellular vesicles are not just involved at the premetastatic niche, as I told you, but they are involved either from the pre-cancer origins, very at the beginning, during the formation of the primary tumor, in metastatic colonization, and of course, in response to therapy. This is a review, or somehow a review that you can see the complexity of the application of the different areas of the extracellular vesicles. But I wanted to share with you this slide because it's probably one of the slides that basically captivated me when I was a postdoc in David, because I realized what is the power of the extracellular vesicle, not just sharing material, but actually communicating to other cells. When I realized, honestly, I'm talking about 2008, right? We didn't know much about the implication of extracellular vesicles, but when I was in the laboratory and I was looking by myself how there is a horizontal transfer of material between cells, and actually how can you change the behavior of cells that are recipients to extracellular vesicles, this is actually what happened to me. I was captivated by these ideas. How can you envision this, or how you can transform a tumor cells or non-tumor cells with extracellular vesicles? And in this horizontal transfer, we knew that proteins were there, we knew that RNA was there, but probably they were the beginnings of the knowledge of how molecules can be transferred among cells. At this time, what we know is that tumor cells can secrete extracellular vesicles. They can transfer material among cells, whether it's an oncogenic cell with mutation, it can transfer to other non-tumorigenic cells. Let's say we are studying melanoma and how these affect to melanocytes and along the progression of the melanocyte. We understand also, we define how tumor secretive factors, in this case vesicles from melanoma, can transfer material to bone marrow progenitor cells. In this case, they were host cells, and they were educating them and forming pro-vasculogenic and pro-angiogenic profiles. So for me, it was like shocking, I have to say. I was working and looking, okay, how come I can treat cells and they can behave differently after the treatment, and we can modify the phenotype of these cells just by the treatment. And I was really, looking in this idea. But not only that, at the very last part of my postdoc, and when I started here at the CNIO, I was using also them as a biomarker in circulation, not only in plasma, but actually we published several works about the use in other biofluids, such as the seroma that you take from the lymphadenectomy. In melanoma patient after the surgery, you can take this biofluid that is in the lymphadenectomy. They put a drainage, so I use this drainage as a biofluid to measure relapse. But today I'm not going to talk anything related to liquid biopsy, but I'm going to focus in other concepts. So I just want you to clarify this. Just give you an overview of the influence. We just recently published this review about the influence of extracellular vesicles in the primary tumor growth. We selected several ideas in this review, but I pick up this because, as I told you, is one of the main probably ideas that captivated me when I started, is the horizontal transfer or influence of proteins can be shared out in extracellular vesicles. I didn't put today about the classification in a small, large extracellular vesicles because I guess you guys know this. I mean, you are in the field, but today I am using the general term extracellular vesicles in overall, but I mostly work at the beginning with exosomes. Now, a small extracellular vesicles, so most of the literature is based on that. But if you have any specific question about the EV type, just let me know. At this time, what I can tell you is EGFR was described in microvesicles by Johannes Koch, for example. CMET is one of our work. RAS was described by RAC. So the idea is whether several oncoproteins were shared out by tumor-derived extracellular vesicles, and they can promote the transformation. Basically it's an in vitro transformation, but also in vivo of other cell types, and whether this transformation is real, whether these materials are actually influencing the behavior of the tumor, or are we only shaping the phenotype of those non-tumoral cells around the tumor. This is one of the main, let's say, interesting ideas in the field that, as I told you, they captivated me at the very beginning. This transformation through extracellular vesicles. We know also that there is also activation of other cell types such as CAFs through smaller RNAs. There are several other indicators that can promote epithelial to mesenchymal transition, although EMT normally, as Elena mentioned, I worked in EMT time ago. At this time, there is no like a really strong work or strong data in how this could happen in vivo. In vitro, there are several reports telling that EMT can be induced by extracellular vesicles, but whether this is happening in vivo, and this is a matter of discussion that we can discuss today, whether these extracellular vesicles can promote those changes in vivo, and how should we approach this to understand the complexity in vivo is the real frame that we really need to work at this time. I think this is one of the critical areas that I would like to share with you today. We really need to understand what is the role of extracellular vesicles in vivo. This is challenging, but at the same time, I think it's necessary to move the field. At the very end, also we have the ideas that EVs can promote immunosuppression. PD-L1 is shared out in extracellular vesicles. It can promote exhaustion of T cells. And as you know, it can modulate the behavior of immune cells. Whether this is happening again only strictly through extracellular vesicles from the tumor, or maybe other cell types in the primary tumor can promote this exhaustion, it is a matter of study. Based on the role in metastasis, I'm going to talk mostly of the premetastatic niche, but you should be aware that there is also a lot of studies going on in the potential implication of immune cells at the premetastatic niche. David Leiden is working into that as well, whether these extracellular vesicles promote changes in immunity at metastatic niches, and this could be a balance of pros and cons in signaling. The idea after this year, and this is an overview of what we published with David, we published this work that was the initial study that I did with David about how extracellular vesicles can educate bone marrow-derived cells. Later on, we published the implication in metastatic organotropism, the formation of the premetastatic niche in the liver with Bruno. Also, Gonzalo published this about the premetastatic niche formation in the brain, and we have recently published this in Nature Cancer about the implication in the premetastatic niche in lymph node locally. So overall, during this year, we defined the role of extracellular vesicles in the premetastatic niche in distal organ, but also in the lymph node, giving an overview of the power of these vesicles in vivo, and whether this is happening also dynamically. This is one of the limitations of our studies, was that we were injecting, as you probably know, those extracellular vesicles. So this is why I'm telling you that we should approach this now more from the context that we should understand whether these vesicles can promote these interactions in vivo in a dynamic system. These ideas were actually completed, but other works. I'm not going to really go into details in every specific organ, but after our original publication, there were several others promoting different molecules at the premetastatic niche formation in the lung. The same happened in the liver, for example, by the implication of small RNAs. Again, in the brain, there were several small RNAs implicated. And finally, there were also several reports telling us that extracellular vesicles can modulate immune cells and immunity at the lymph nodes. But overall, the idea and the summary is that during the premetastatic niche formation, what we know is there are mainly 3 events that will happen, and we call it like a hallmarks of the premetastatic niche. One is vascular leakiness, as I told you. If you remember, this is happening in the premetastatic niche formation without extracellular vesicles, but we know that extracellular vesicles can promote vascular leakiness. They promote also extracellular matrix remodeling, and finally bone marrow derived cells recruitment. So these are the 3 hallmarks that we consider that extracellular vesicles can impact in the premetastatic niche formation. And whether this is happening, in vivo and in the, let's say, in the real life, this is a matter of study. But I wanted to share with you today several ideas, as I told you, in order to see how to mimic the delivery of extracellular vesicles at the premetastatic niche. We really need to understand it when, right? Understanding your time course, where, or to define this, what we call the physiological dose. Probably you guys are under reviewing several papers. Most of the questions are, what is your physiological dose, right? We are going to discuss this, and how, determine this cargo responsible for this effect. The when is, of course, you really need to understand the course of the metastatic disease. As you know, metastasis could happen through lymph node, but could happen also distally through the vascular system. So you really need to understand your model. And in order to understand your model, you really need to investigate whether your tumor is metastasizing through lymph node or distally through the vasculature. You probably know, Stephen Paget, that, the seed and soil metastasis were only, in this case, there was a hypothesis whether metastasis is not happening by chance, but only in a specific organ, because you have to have the seed, which is the tumor cell, and the soil, which is a specific microenvironment. This was challenged later by, James Ewing, telling that the dissemination occurs just by mechanical factors. And the reality today is we have a mixture of both. And this, again, Joan Massagueira, Isaias Fiedler told us the reality is whether, of course, the seed is important, the soil is important, but also the mechanical forces are important, because this is a matter of combination of everything. And this combination actually ends up with the metastasis in a specific organ. That could be brain, could be lung, could be bone, depending on the 3 factors. The seed, the soil, and the mechanical forces are important into that. And this is what we know now. Several studies, of course, define intrinsic factors that are involved at the, metastatic dissemination are important. Probably, in brain metastasis, you have HER2 amplification, for example, you will likely have more prone to metastasis in the brain. And during these years, we've been studying not only the intrinsic factor, but also the extrinsic influence of extracellular vesicles in the metastatic formation. And this is part of the work with David. We published that integrins can play a active role in the metastatic dissemination and metastatic organotropism in breast cancer, for example. But besides that, and understanding how your metastasis is happening, is to understand your tumor model. And I put several slides that they are all, because this is probably from my first study. Understanding your model is really important in order to know what is the time course that you are using. For example, if this is melanoma and you use flank injection, we started by injecting extracellular vesicles. You see these arrows. This is where we started injecting them, and we were observing that those metastases will be happening around 28 days, right? And this is, the time point when we finally have the end time point. But if in order to study the premetastatic niche, we were studying this around day 18, and this is exactly where these tiny cells are homing, and the V16 and cherry cells are getting there right at the day 18. And this is to define your window of the premetastatic niche is really, really important. I spent probably 3-4 months just trying to understand my premetastatic window in this model. So this is why I wanted to share with this. This is probably my year 1 as a postdoc in David's lab in order to define this premetastatic niche window. And of course, they will evolve to metastatic behavior later on. Also, it is important to understand the limitation because the injection route where you inject the extracellular vesicle, for example, matters. If you inject intravenously, you are likely targeting lung and liver. If you are injecting intracardia, you are getting extracellular vesicles to the brain and the bone. If you inject intrasplenic, you will go to the liver. If you inject foot pad, you will go to the lymph node. So this is, of course, first, you really need to understand the organ that you would like to study. But in order to mimic the premetastatic niche formation, it is crucial for you to understand the delivery of your extracellular vesicles. And this is important based on what you see here in the tumor model that you study, because it's not the same if you are going to study premetastatic niche in the lung than in the liver. So for me, taking the time to understand my model actually was several years until we decided where to inject and when to inject those extracellular vesicles. At the end, of course, you will also have to select the delivery method. And in this case, it's not just the delivery, but labeling those extracellular vesicles. You have different also approaches, which is you can have membrane dyes, which this is an odyssey, by the way, that you can use for your Western blot. So I decided to use the odyssey just to look extracellular vesicles. And we were doing Western blot in the lungs of those mice. So this is one lobe of the mice, the other lobe, and all these were extracellular vesicles injected, and they were labeled with membrane dyes. They are near infrared dyes. The problem of those dyes is, as you know, is aggregation. Of course, these are not small extracellular vesicles. This is clumps probably in the lungs, and the sensitivity, because believe me, if you see here, there is nothing to do in sensitivity, for example, with if you use radioactive labeling, that you can really see probably inject of 1 microgram or even less micrograms of extracellular vesicles. In here, we probably injected 10 micrograms. In here, we injected less than 10. So of course, the sensitivity of the methods is important, and the specificity as well, because in here, for example, while we use small extracellular vesicles with a fusion protein, so we fuse this NGFR receptor with GFP, we only use extracellular vesicles we injected. You see, these are lymph nodes from those mice. You can nicely see the uptake of small extracellular vesicles. The main problem of this could be time consuming to generate the model, also potential artifact by the generation of the fusion. So I guess my message here is that there is no an ideal method for it, but actually you really need to try what is your ideal method. And the physiological dose, again, I mean, in my case, for example, you really need to understand your model. In my case, we mostly use this V16F10, which you see the ratio of secretion was 1.5 micrograms per million, but you really need to understand your method of isolation, that in my case was ultracentrifugation. The yield of ultracentrifugation is really low, as you probably know. You only get 15–30% of the particles. So the estimation was to get 5 to 10 micrograms of extracellular vesicles per million of cells. And this is just mimicking the injection of 1 million cells. So as you know, there is a standard in the field of metastasis that you inject 1 million cells in the lung, they go in the flank, and you analyze the metastatic behavior. So we try to mimic this physiological dose. I am telling this because one of my main question that I have when I review my paper was that what is your physiological dose? And this is how we rule out the physiological dose. This is important in order for you to reach the review of your work, or whether this is important to understand. After the isolation and labeling, we decided to look at those organs when they reach extracellular vesicles. You see extracellular vesicles increase. They reach the lung within 5 minutes, so really, really fast. But at this time, we look at the delivery in distal organs, and we were able to define the distal organ of dissemination. So this is something that is important for you to understand how to inject, where to inject, how to label, and which organ you should look for the premetastatic information. And all this, as I told you, I basically was working for a year and a half at the beginning of the project just to understand my model. And believe me, this is important. This will be my advice, right? As I told you, this is the end. Sorry, I think this is a double slide. Sorry, because this is, again, I don't know why. So the next challenges we face right now, and this is the last part of the talk, where I would like you to see where are we going in my laboratory right now. It is important to understand, as I told you, the role of these extracellular vesicles in vivo, in physiological models. And I think this is one of the key points that we should all go, that to understand the secretion in vivo and the dynamics in vivo. And today I'm going to give you a hint of this because I am working, but not as there are a couple of laboratories I will show you today that are doing much better work than me in that. But at the end, I would like to give you some hints of the understanding the heterogeneity and the composition and the therapeutic use of extracellular vesicles in the last part of the talk. Regarding the role in vivo and the physiological context, I just would like to share two works that, two groups that I like a lot, and they are colleagues of mine, like Yves Leclerc, who is in Canada, because they actually use this model in order to mimic the delivery of extracellular vesicles in vivo. And they produce this model. It is a turbo GFP protein linked to the methylation of a protein. So you can see a nicely delivery of this protein in the surface. And of course, it will be shared out. And in this work, they analyze in vivo, like injecting in the flank and looking at the premetastatic niche that those EVs are able to reach lungs. And you see day 15 is more or less our premetastatic niche. Starting in them is day 10, day 15, day 20, day 25. And this is important to understand. This is what we were mimicking by injection is nicely get it by in vivo model. I think we should try to get to the in vivo approach. Whether this is possible or not is important to approach from your, from your experimental model. And also, I would like to share with you this work by Jackie Goetz that he's studying this in therapies, and I would like just to see how this group is injecting extracellular vesicles and looking at the interaction of EVs with this dorsal aorta and the flow, actually the real flow of extracellular vesicles in this. And I put here like let me share with you like a video, because it's really interesting to see whether these extracellular vesicles can go in the flow of these mice, right? This is like basically erythrocyte and the extracellular vesicles are getting there. And in this work, they see these areas where these extracellular vesicles are clumping are actually areas of macrophages taking up the extracellular vesicles. And this is one of the things that I believe we should work the most within the next years. So maybe you guys are already doing that. I will be happy to hear your approaches and whether to understand the delivery in vivo of those extracellular vesicles. It is important to understand the dynamics, right? It is as well challenging. So it's not easy to study this in vivo. And this is why we started injecting extracellular vesicles. And in the last part of the talk, I would like to give you a couple of hints of what we are doing based on trying to understand the heterogeneity of extracellular vesicles. And this is because, of course, you normally need to choose your biomarker of a study. And in this end, you can choose nucleic acid, metabolite, lipids, or protein. And in this case, we have a project in the laboratory looking for what we call microproteins, like small proteins in the extracellular vesicles. We started this around 4 years ago. And giving you an overview, what is a microprotein? These are proteins with less than 100 amino acids. They are evolutionary conserved. They are very, very few functionally characterized. In other words, I would say that this is similar to what we knew about microRNAs once it all started. So microproteins, there are many. They are codified by a small open reading frame. They were supposed not to be codified, but actually they are codified by a small peptide. So we don't really know their use or their role in biology. We are trying to understand in biology, in tumor progression, in reprogramming, which is the project that we are doing with Maria Bad, but also in this case, the shedding in extracellular vesicles. I will give you an overview. I don't want to get into the details, but just give you an overview what we are doing and how we are approaching this. We are starting with pancreas. This is the model of a study. we are getting tissue explants from primary tumors from humans, right? And also we are getting these explants, or what we have either is healthy and non-healthy tissue, with tumor and healthy tissue, and analyzing by putting those explants in vitro overnight. We take the supernatant, this supernatant we put it through mass spectrometry, and then we analyze the microproteins. For that, you really need to actually develop a mass spectrometry database. And this is what we did during the last year. We developed a database of microproteins, and then we analyzed the microproteins shared on extracellular vesicles. We are doing the same approaches with plasma. As you see, there are small numbers, but still quite interesting results, as you will see now. From plasma, for example, what we start to analyze is this analysis by mass spectrometry. We normally identify many. You can imagine, these are only 124, but we identify 1,000. The main problem of this project is to actually know that your microprotein is real, because they are undefined. We don't know anything about them. So you really need to put really specific annotations and be really strict in the selection. And we end up with 83 potential microproteins secreted in our database, and they are mostly located in the 3' UTR of these genes. And as you see here, this is just the distribution. It is interesting so far. I don't have data to share with you today, but what I can tell you is the distribution, as you see here. Even if you go for canonical proteins, or you go for microproteins, in this case, you can find specific signatures that are only in the PDAC or only in the healthy donor. So what we are doing right now is to really define these signatures. And the idea of this project is, besides defining the signature, of course, getting biomarkers that can be identified in the plasma, but not only in the plasma, but also in the tumors itself, as see the biology behind. And for that, what we are doing is another approach using these explants. As I told you, the explant, we put it in vitro, we take the supernatant, we isolate extracellular vesicles the next day, and then we do the mass spectrometry analysis. You see here, we end up with a pretty much amount of proteins, and then we really need to get rid of the potential false positives. So we end up with 23, then we clone it, we express it, and we saw only 13 of them detected intracellularly, and out of them there were only 3 secreted in extracellular vesicles. So as you can see, it's a time-consuming project, but you end up with potential candidate. We end up with one of them that I'm going to show you today. We call it 13 because it's the 13th analyzed. The idea here is, when you overexpress this candidate, for example, in the BXP3 model, which is a pancreatic cancer cell, you see that they can increase intrinsically the metastasis, but also if you express it in another model, MIA-PACA, you increase metastasis, in this case in the liver, which is a typical metastasis for the pancreas. And the idea is to see whether this could be functionally functioning not only inside of the cell, but also in the extracellular vesicles. And this is the working model we have right now, is that we believe the 13 is secreted in extracellular vesicles. They actually are having a role in the primary tumor, but we believe this is important also, or the idea is to look in the future to if they are also playing a role in the premetastatic niche formation, or even respond to therapy, because these are highly aggressive tumors. So this is a project I would like, I wanted to share with you today. And in the very last one that I would like to share is the one that, just to give you an idea whether the information in extracellular vesicles is not just useful to understand the biology in the premetastatic niche formation and in metastasis, but also it can be used to define new therapies. I call it extracellular vesicles inspired therapies. As you know, you can get specific markers in EVs. You can use it as a potential therapeutic agents. And for that, you really need data that support this therapeutic application. So we are studying in this project the effect of natural killer-derived extracellular vesicles, natural killer cells-derived extracellular vesicles. As you know, natural killer are driving the behavior of many immune cells, right? They promote or actually T cells maturation, and they promote either the killing, direct killing, but also the regulation of anti-tumor immune cell responses. So we believe that if you engineer specific vesicles to be a natural killer derived extracellular vesicles, you would be able to potentially control the immune system. And this is inspired in a work by, probably you know, Dr. Sanchez Madrid here in Spain. They published this paper about the analysis of natural killer derived exosomes, and they saw specific microRNAs that are secreted in those natural killer derived exosomes, and they can modulate the behavior of T cells and also dendritic cells. So inspired in that, we wanted to analyze the role of any specific microRNA loaded in nanoparticles. So we are engineering novel nanoparticles with these different microRNAs in order to modulate the immune cell system. This project is basically at the beginning to look the biodistribution of these nanoparticles in vivo. So for example, we inject those nanoparticles with different microRNAs, we see the biodistribution in different organs. What we can see, for example, is that here is an odyssey. In red, you see background. In green, you see nanoparticles. And the thing is, this is controlled. These are nanoparticles without any microRNA. These are nanoparticles with RNA. The crucial thing here is you don't see any difference. As you see, there is no difference if you inject in the foot pad or systemically, whether those particles can go to the tumor or the liver. In other words, we couldn't see any specific biodistribution of the nanoparticles if you inject in the foot pad or systemically retroorbitally in the tumor or in the liver. Okay, this is life, this is science. But luckily, we could see that those nanoparticles were retained in the foot pad of those mice who were injected in the foot pad. Sorry, we looked them in the lymph nodes of the close to the foot pad. So in other words, we were able to retain nanoparticles after foot pad injection in the lymph node of those mice who are actually the organs that we wanted to be retained, because we are thinking to modulate the immune cell behavior. So starting on this and verifying that nanoparticles can be retained in the lymph node, we started a project in order to see what is the therapeutic response in tumor-bearing mice. And this is the project that we are doing right now. We are using metastatic models of the B16F10 and also the JUM, for example, that is immunotherapy resistant. We are injecting in the flank of those mice tumors, and we are treating with nanoparticles in the foot pad in order for those particles to reach the lymph node, with the aim to reprogram immune cells in order to be anti-tumor, right? So we started the project. We have pretty promising data that I'm going to share you with today, three slides. One is that we have a couple of candidates, microRNAs, that if you inject in the foot pad those particles, we are having an anti-tumor effect. We have a couple of them. One of them could be, working nicely to reduce primary tumor growth, but actually the idea is not just to promote anti-tumor growth, which is important, of course, but we would like to combine with immunotherapy, and this is why we are using this model, which is a JUM 1.1, which is resistant to immunotherapy, as you can see here, was published, and we are trying to improve the effect of anti-PD-L1 with these nanoparticles. We have promising results. Still, this is a year 2 in the project, so I decided to share this because Barbara asked me to share these ideas, but it is still promising project, but still early project, that we believe that if you load correctly specific microRNAs in your nanoparticles, you are able probably to promote anti-tumor cell responses and probably improve immunotherapy treatment. The idea of this project is that you are able to load a specific microRNA in order to mimic physiological responses. At this time, we are looking at the extracellular vesicles from natural killer cell, but of course, you can imagine this in any of your projects. Imagine you have a specific microRNA from macrophages or T cells, or even tumor suppressor microRNA, metastasis suppressor, or you want to manipulate what we call aptamers, or boosting nucleic acid, you can engineer. Where we are trying to go is to develop extracellular vesicle inspired therapies to use as anti-tumor therapies. So this is the idea of the project. I hope that with several years more, we will be able to define the role of this microRNA and whether T cells or specific T cell responses are regulated. The last slide I would like you to share with you today is this one. I was mentioning before that in Spain we have this Spanish group of research in extracellular vesicles, JVEX. This is the board. I belong to the board. You probably have your own in your countries, but of course, the main benefit to be here is, at least for you guys, is that becoming of this network, you are able to use this MOVE initiative. Right now, we are also having some initiative with South America as well. So we are engaging not just research in Spain, but we are engaging people that would like to spend part of the time in Spain, and actually our fellows that can go to other countries. So this is, I would like you to have the webpage in case you are interested. You can, of course, get into the details. And the last is, I would like to thank my people here at the CNIO. As you see, really, young people, probably as you guys, with really aiming to understand the research of extracellular vesicles in cancer. And I would like to thank them all, funding sources, and you guys for your attention. Thank you so much.
Thank you. Thank you so much for sharing your experience, Professor Peinado. On behalf of the SNEV ECM, we really appreciate your participation here, your insights, as well as your future work and the initiative that you shared with us. So now we have some time for questions. Please feel free to send your questions, message, tabs in our chat box for transmitting them to our speaker. So to start, we have some comments here from Vic Jurevich. He said, would it be feasible to perform ribosome profiling on the primary tissue for S-ORF identification? ncRNA-derived MIPs could be more tumor-specific than those previously described.
Actually, yeah, indeed, this is the best. I have to say, Big, right, is the name. This is the best approach you can do. Actually, the one you are proposing is the one that we are doing right now. This project started, as I told you, 4 years, 3-4 years ago, which we didn't know much about how to approach it. So we did mass spectrometry in small proteins. But this is not, even if I did it, is not the best approach. The one you mentioned is the one you need to do in order to be sure 100%. Because, of course, with the ribosome profiling, and if you do this, you are 100% sure that your protein is being codified. And doing this, you are 100% sure that your protein can be expressed. And then you can match the small extracellular vesicle with that. Totally right, 100%. I would do that, and we are doing that, actually.
Cool. Thank you. Now we have a subsequent question also from Vic. He said, have you detected mRNA matching the S-ORF products in EVs?
Well, we have detected the small open reading frame in the EVs, but how we did it is a trick, meaning that since we were doing mass spectrometry to identify this peptide, what we did was overexpressing the peptide with a fusion with HA, right? But in order to design a specific probes for mRNA, was not that easy to find for all the different, let's say, candidates we have. So in other words, if you have a candidate that allows you to specifically target this small open reading frame, we did it. But there were many that we were not able, because since they are open reading frames, they could be overlapping with other parts of the other open reading frames. So we were not sure about these approaches in several others, so we clone it and overexpress it. But theoretically, you could be detecting this in the extracellular vesicles and also in the, those cells who you treated.
Awesome. Thank you, Professor, and also thank you, Vic, for the questions. Now we have questions from Delaram Dorot. She said, thank you, Hector, for the fantastic presentations and valuable research you have conducted. I wonder, how can we address the potential for EV-mediated drug resistance in cancer therapy, and what therapeutic strategies can target EV release or uptake?
So regarding, I mean, I'm not an expert in therapy resistant and drug resistant. Most of the studies I was reading that how the people is normally approaching this, try to confer resistant to cells that are not resistant. So you take extracellular vesicle from resistant cells, and then you treat non-resistant cells in order to promote the resistance. This is one of the approaches that I saw in the literature, and this is normally how the people try to do it. Whether this is depending on the specific model, whether your molecule is the trigger of the resistance, the model they normally do is knock out the specific candidate from your model and see if the candidate, your vesicles derived from the knockout system, are not recovering the phenotype. This is how what I read is the most. Of course, there could be many, but honestly, I'm sorry because I'm not an expert. I'm not doing this. I'm doing therapy resistant, but not using extracellular vesicles. But I mean, I guess this is one of the approaches. And regarding the strategies to target EV release, one of the main strategies is, as you probably know, knocking out RAP27A, which is one of the main drivers of the extracellular vesicles release, like the multivesicular body. This is one of them. The other is using this GW is, sorry, I don't remember the number. The sphingomyelinase inhibitor. The idea is whether you can modulate the phenotype of those cells to be reduce the secretion, but not killing the cell, because I can tell you that when I started knocking out RAT27A, we saw that there is a deleterious phenotype because the cells start to die when you knock out. So it is a balance of physiology. You have to have in mind that EV release is important for physiology in the tumor cell. So if you knock out RAT27A, of course you can prevent release of EVs, but you can promote cell death. So this is a matter of balance. GW can be used, but there are many other inhibitors of release of extracellular vesicles in the market that you can try. But GW is probably the most. The thing is whether this is strictly specific of small extracellular vesicles. This is not clear. It could be also affecting large because it's a sphingomyelinase inhibitor. I'm sorry, I only have the last part is about the uptake mechanism. Regarding the uptake, again, it's not 100% one mechanism, but there are reports telling that it could be endocytosis, it could be macropinocytosis, and some others are telling that it's receptor dependent. In my case, when I knock out integrin, for example, I think it was integrin beta 5, I think, no, alpha B, I saw, we reduce the uptake by 50%, but not 100%. So integrins are important into that, but not 100% important. I say 50% important. There are additional mechanisms, probably endocytosis and macropinocytosis, that are involved in that. Whether you have those inhibitors, that could be important.
Thank you, Professor. You already gave some hints regarding the EV heterogeneity, and it can have the option to move forward to the next question from Delaram Doro, where she said that regarding your opinion, how can we design EV-based therapies to selectively deliver anti-cancer agents to tumor cells while sparing normal cells, given the overlapping EV uptake mechanisms?
Sorry, can you repeat that? Sorry, who was this?
Delaram Doro. How can we design EV-based therapies to selectively deliver anti-cancer agents to tumor cells?
Yeah, I mean, this is a really, it's really hard. I mean, simplifying is a hard question because in my opinion, at least in my experience, engineering the complexity of extracellular vesicles in vitro, this is what we are doing right now, right? We are engineering a particle in order to deliver the small RNAs. but we cannot really reproduce the complexity of the in vivo delivery of extracellular vesicles. In other words, probably it's not just one molecule, but a multi-molecular interaction on surface. Integrins are important, but as I mentioned, for delivery, but probably not sufficient to have the complexity. There are many people asking me, can we target a specific particles with molecules on the surface in order to be lung-tropic, liver-tropic, as to use as a therapy? My answer is not that simple to reproduce, and I couldn't reproduce myself this. So honestly, getting for therapies anti-tumor to be specific in a location at this time, I think we are quite far from there right now.
Yeah, okay. Thank you. And regarding normal cell-derived EVs, how is the balance between pro and anti-tumor effects of these EVs from different sources?
Yeah.
Is there any reference or idea?
No, I mean, it's a quite complex, I don't know if you've been in last year, I was in ISAP, right, giving this concept that, of course, along the evolution of the tumor cell, there are many changes, but extracellular vesicles change as well. There is an evolution of the phenotype along tumor progression. And this is a balance of tumor and anti-tumor properties, but not only of the extracellular vesicle, but the stromal cells and the complexity of the interaction with the microenvironment. Whether there is a balance between pro-tumor vesicles and anti-tumor vesicles, the obvious question is yes. How can we approach that? Honestly, it's overwhelming because at this time we are based mostly in cellular responses. So if you use a T cells again, the tumor cell will kill it. Whether you can use extracellular vesicles from T cells, CAR T cells as an anti-tumor therapy, and whether tumor cells will react secreting more vesicles to counterattack, as you see, this is much complex. So the theory in this question is true, meaning that I guess it's a balance. How to approach this complexity is honestly overwhelming to me at this time because it's based on my experience. I couldn't control the complexity of the secretion of extracellular vesicles, not only in my system, but if you compare my system with breast cancer, is radically different. For example, melanoma is totally different. So I guess there is a balance in these pro and anti-tumor properties. Whether to control it, I guess, is the question, right? To understand this complexity. It's not to be negative. It's just, based on my experience, it's just complex.
Yeah, thank you. Absolutely. I think that it is an open field to be studied.
Yeah.
Even we can find differences between subtypes from the same tumor, right?
Yeah, totally. I mean, in my experience, for example, I mostly work in melanoma, so we work for less melanocyte, right? Low metastatic melanoma, high metastatic melanoma. You do proteome analysis radically different. I focus in the top protein that was secreted in melanoma, c-Met. I knock it out. I reduce metastasis, but there were others, such a complexity that they were influencing the behavior. So we were not blocking metastasis. We were slowing down metastasis. So the complexity of the composition is, again, overwhelming. It's just, of course, in our publication, we mostly focus in one gene that is the top one, but there are many other, probably 20 or 30 more that can allow these vesicles to home and to execute the system.
Yeah. Thank you. So moving a bit from the biological space for the ethical and society space, we have a comment regarding ethical and practical constraints. As soon as we already realized it regarding the heterogeneity of these samples, it suggests to obtain more and more primary tissue from patients. Yeah. However, it can be especially considered as a bottleneck alongside ethical considerations for invasive biopsies. How can this be justified?
I mean, of course, ethical protocols are right there. We really need to take in consideration everything. Everything. In Spain, we have a really strict ethical protocol. The idea, at least in my research, what we are using is extra tissue. So in order, Of course, if you are telling me that you really are doing this just to get extracellular vesicles, I agree with you that maybe you cannot justify this. However, in my approaches, the biopsies we are receiving is normally from extra tissue after surgery. So those patients were getting resection of the primary tumor. They have they have a stage probably 3, 4 pancreas PDAC, right? And we were getting explant from this tissue. However, we are approaching right now, this project is coming closer to the stage 1. Of course, getting in not only because you cannot catch it probably stage 1 pancreatic cancer, but whether to justify getting a tissue from any pancreas, I don't think, as you say, that this is justifiable. However, we are approaching that from the liquid biopsy part. So instead of getting tissue, we are getting plasma from stage 1, or probably 2, and also earlier stages and non-malignant stages from plasma of those patients. And again, we are following the rule of the ethical issue, meaning that this plasma is taken anyway from the patient in order for following up the patient. So I agree with you. Getting just tissue for these specific approaches, it could be fishing expedition, honestly. Getting extra tissue from biopsies that are already performed or plasma that is already taken, this is how we are normally approaching that.
Yeah, thank you, Professor, for these insights and these comments. So now we have a question from Rambot Nick Bakach. It says, thank you for the great talk. Considering all the studies currently being performed on differences between EV isolation methods, why UC ultracentrifugation is still your method for choice for EV isolation?
Yeah, that's funny because I mean it is published and we mostly started, as you see in our publication, ultracentrifugation, right? It's a standardized protocol that we've been publishing during the probably papers that I showed you. However, right now we are changing this because I agree with you that things are moving, and we are using right now 3 different approaches more. One is sucrose gradient, so we do ultracentrifugation followed by sucrose. We do site exclusion chromatography, and we are using also Exodus equipment in order to isolate from plasma. So I am not only using ultracentrifugation, probably the methods I showed today use ultra, but we are moving forward to the analysis. And I guess, of course, this is based on the technical or experimental explanation is that we approach this in order to have the higher yield of extraction in extracellular vesicles at the beginning. But now with site exclusion chromatography, we know that we can have even a purer population of extracellular vesicles with increased yield. So honestly, we were evolving. Probably the data we show we've been publishing was mostly ultracentrifugation, but we are changing this behavior.
Thank you, Professor. We have more questions regarding the doses, the dose you use to represent the physiological conditions, set as 10 micrograms of protein of EV per million cells. My question is that was the dosage achieved in total after all 5 to 6 EV injections that you had? Or do you have an estimation of the particle number that this dose equals to?
Yeah, well, I don't have in my mind the number, but I can tell you my view. My view, if we inject 10 micrograms probably twice a week during 3 weeks, you will have 60 micrograms, right? The primary tumor, the first day we inject in a mouse, right, is 1 million. So in the flank injection in melanoma, you inject 1 million. It takes 7 days to reach probably like a small tumor, which is definitely more than 1 million. Let's say 10 million. This is taking up to 28 days in 3 weeks, which is a tumor that is 1 centimeter cubic, right? Which is really much bigger than 1 million and much theoretically secreting much more than 60 micrograms. So we cannot do math with biology. We are mimicking the constant secretion of 1 million the equal amount of extracellular vesicles to 1 million every 2 days a week, let's say. And this is a constant rhythm. It's like when you have a pump. But the dose that we are using with these extracellular vesicles is definitely much less than the dose that is these mouse are receiving probably in the last week of age, which the tumor is almost 1 centimeter square or cubic. It is it's probably a ball generating extracellular vesicles. So even if we cannot do much, I think that the amount of vesicles that we are injecting is equal to the first point where we injected this 1 million. So I guess the dose is less. However, I can agree with you that even if the dose is less in number, is shorter in time, so which is something that we really have to. I am being autocritic. Of course, you are injecting the EVs from 1 million cells in 1 second, right? This intravenous injection you shot is probably, this shot is much stronger than the delivery, continuous delivery of EVs through the whole time. So honestly, I don't think they can be equal. We are getting a model system, but I agree that can be criticized that you just load this in 1 second and maybe those is not the same in vivo. I guess we really have to have a balance on whether to have the biology or the numbers, and the best system honestly is what I mentioned to you, trying to get to the in vivo scenario and develop system that can help us to mimic the delivery in vivo. This is honestly what we need to do. I mean, this is what we are working.
Awesome. Thank you, Professor. I think it is clearer now. So here we have a final message from Dilar on the road. She said, "Thank you, dear professor, for the nice presentation and replying precisely to the questions. It was very useful." I think that we share this thought. It was a really productive talk. And thank you so much. I don't know if you, Elena, would like to add something, or you, Professor, do you want to give any final messages from this talk?
No, I would like, first, again, thank you guys for, for the discussion. Thank you guys for your interest. And of course, if you need anything, just reach me. Feel free to reach me directly. Any idea that you may have. I think you guys are the future of the field. So I hope these ideas are helpful for you. It could be from the critical point of view, the ways to improve how to approach the biology of extracellular vesicles. And I hope my words and my work could be, inspiring you guys. And in 10 years, I will see you giving this talk away. Thank you really, really for your time.
Thank you. Thank you so much.
Thank you so much.
Thank you.
So I think it's everything for today. Thank you for being here, and see you in the next talk. See you. Have a nice day, morning, afternoon. See you.
Bye, guys.